will be dealing with the secondary growth. In secondary growth we will be talking about the vascular cambium, its activity, and we will be talking about the heartwood, sapwood. We will be talking about the formation of annual rings, lenticels, periderm, all these topics, periderm formation, lenticels, secondary phloem, secondary xylem. So, when we talk about the secondary growth, we will be dealing all these topics here. Now, what one by one you will get into detailed study of this type of headings. Okay, we here with as per your syllabus, we will be discussing with reference to two structures here. We will be discussing first about the dicot stem, and again we will be discussing about the dicot root. Here in as we all know, the normal secondary growth can be seen only in the dicot stem and root. We do not see the existence of the normal secondary growth in the monocot root or monocot stem or monocot leaf or anything related to monocot. So, we will be studying in detail about the normal secondary growth in the dicot stem. So, in general when we are studying the structure or vascular tissue organization of a dicot stem in normal growth, what is the vascular bundle here? As we studied earlier in the TS, the vascular bundle will be conjoint, collateral, open and endarc. Conjoint, collateral, open and endarc type of vascular bundles are present, and in between the parenchyma cells which are present, the parenchyma cells which are present between the cambium are called as intrafascicular cambiums will be seen. You will see the presence of intrafascicular cambium. Presence of intrafascicular cambium is seen in between the medullary rays. The parenchyma cells in between the medullary rays are called as intrafascicular cambiums. Now, the medullary rays, the parenchyma cells of the medullary rays which are located in between the interfascicular cambium, in between the fascicular cambium are called as the parenchyma cells in between the medullary rays are called as intrafascicular cambium, and the parenchyma cells or medullary rays which are present in between the intrafascicular cambium, are called as the interfascicular cambium. Now, altogether this, see here, this is called as fascicular cambium, and this in between the two fascicular cambium is called as interfascicular cambium. So, there are two types of cambium formed here, interfascicular and intrafascicular cambium is formed here, and here this interfascicular cambium becomes meristematic and develops a potential of continuous division, and they formed into strips of interfascicular cambium. Later on, this inter and intrafascicular cambium will continue mixed together and form a continuous ring like structure. Okay. They collaborate with each other as the multiplication or division potentiality increases. Both will become meristematic and they undergo a continuous ring formation. So, you can see as in the diagram here, a ring of vascular cambium is formed with a combination of both inter and intrafascicular cambium. Because of this formation of ring, there are two types of initials are developed. Those are called as this both inter and intrafascicular cambium divide and form into a ring of cells. So, this ring of inter and intrafascicular cambium forms a continuous ring of the vascular cambium. These cells of the ring will give rise to two types of initials. These are called as fusiform initials and ray initials. Okay. Now, what are these fusiform initials and ray initials? Now, first we will talk about fusiform initials. So, in fusiform initials, okay, fusiform initials, we are talking about fusiform initials. These are the initials which are elongated and spindle shape. These are elongated and spindle shape in structure, and sometimes they may, here these are elongated and spindle shape structure. These fusiform initials may be arranged in either in regular rows in some cells it may be arranged in regular rows, then this type of fusiform initials which are arranged in regular rows, then these type of fusiform initials are called as stratified cambium. Those are called as stratified, or it can also be called as if the fusiform initials are arranged in regular rows, it is also called as stratified or storied cambium. Okay. And one more, this is one form of fusiform initials. Same elongated and spindle shaped fusiform initials, if they are not arranged in regular rows, irregularly arranged, then those are called as non-stratified or non-storied cambium. So, fusiform initials are those cells which are elongated and spindle shaped, and if the cells of fusiform initials are occurring in a regular ring like or in a regular row fashion, those are called as stratified or storied cambium. And if that fusiform initials, in some cases they are occurring in irregular form, the regular row formation is not there, then that type of cells is called as non-storied cambium or non-stratified cambium is present. For example, this here you can take an example of okay, this can be taken an example of all the cambium and the xylem and phloem tissue which are arranged or which exhibit a non-stratified condition in the case of the gymnosperms. Okay, this is about fusiform initials. Now, when we come to the second one, that is ray initials. Here in ray initials, the cells are isodiametric, and this will give rise to the presence of secondary medullary rays. Okay, where in fusiform initials the cells were not isodiametric, in ray initials you will see the presence of isodiametric cells, and this will also lead to the formation of secondary medullary rays. Now, when we come back to the ray initials, or if you are talking in terms of fusiform initials, next when we come to when we come to the fusiform initials, there are n number of periclinal divisions taking place, because of which there is a growth of secondary phloem and secondary xylem is occurring, and here this may also lead out of these cells formed, secondary xylem and secondary phloem, there are two types of cells existing, two types of cells are formed. Out of these two cells, you will see one will be in meristematic condition and the second one will develop either into xylem initials or phloem initials. Fusiform initials will undergo a number of periclinal divisions and will give rise to secondary xylem and secondary phloem. There are basically two types of cells existing which may give in the division of the vascular cambium or in the formation of vascular cambium. Of the two cells, one will be meristematic and the second one will become the xylem initials or phloem initials. For example, when the division occurs, the formation of the vascular cambium, when the division occurs in the cambium, if one of the cells is meristematic, then one inner cells, out of the two cells, the inner layer of the cell is meristematic. In the division of the cambium, out of the two cells, if the meristematic cell is considered as the one of the cells, then the one layer will become as xylem initials and it will be towards inner side, and if the meristematic tissue is considered as the outer layer, then the second initial will be, then it will be the phloem initial, and this will be outer. When meristematic tissue is towards the outer side, it will lead to the formation of xylem initials. When the meristematic tissue is towards the inner side, then it will lead to the formation of the xylem initials. Okay, here where during this cambial division occurring, there is more number of secondary xylem produced than the secondary phloem. In the similar way, when the division occurs, as long as there is a growth of the secondary xylem, it will push the primary xylem towards the center, and when there is a growth of secondary phloem occurring, more division leads to the formation of secondary phloem, and as the secondary phloem comes, it pushes the primary phloem towards the outer surface. So if it is a primary xylem is pushed towards the center if there is secondary xylem coming, and if it is a secondary phloem coming, it will push towards the periphery. Okay, when there is activity of the cambium going on, because of which there is a growth or existence of secondary xylem and secondary phloem. So when secondary xylem is coming, it is helping in the pushing the primary xylem towards the center and secondary phloem towards the outer side. When it is the secondary phloem coming, it pushes the primary phloem towards the peripheral region. Ultimately, what will happen, both this primary phloem and primary xylem will get crushed together, and during the secondary growth you will see only the formation of secondary xylem and secondary phloem. Okay. Now, in case of ray initials, as we talked about the periclinal division in the fusiform initials earlier. Now, if you take the ray initials, it also undergoes n number of periclinal divisions, and it will lead to the formation of parenchyma cells both on the inner and outer side. This ultimately leads to the formation of a structure called as secondary medullary rays, and this also lead to the formation of secondary vascular tissue. Okay, ray initials which undergo periclinal divisions and give rise to n number of parenchyma cells, or it may lead to the development of secondary vascular tissue. In addition, in secondary vascular tissue, it develops parenchyma rays in between the vascular bundles. Those are called as secondary medullary rays, and the water and food storage materials are transported radially through these medullary rays. Okay, now here we come across two terms, those are called as xylem rays and phloem rays. Okay, the xylem rays and phloem rays are the medullary rays which are present are formed from the xylem initials, those are called as xylem rays, and the medullary rays which are initiated from the phloem initials, those are called as phloem rays. As we said, there are two types of initial when there is a division in the cambium. One cell will be meristematic and another may be either xylem initial or phloem initial. So, this xylem initial with this kind of ray of medullary ray, the portion of the ray in the secondary xylem initial, then that is called as xylem rays. And if the portion of the ray with that of the secondary phloem initials, then that is called as phloem rays. Okay. Now, we will talk about the term called as annual rings. What are annual rings here? Another topic we have is okay. Now, what are annual rings? As the name itself indicates, annual means every year you will get the formation of rings. So, year-wise rings are called as annual rings. So, what are year-wise rings here? Now, we are talking in terms of secondary growth. So, we are talking in terms of secondary xylem. So, every year in a plant you will see the development of secondary xylem. Okay. Every year you will see the growth of secondary xylem. Year-wise layer formation will occur in the secondary xylem. So, for example, if it is the stem, okay, this will lead 1 year, 1 secondary xylem, second year, third year, fourth year, fifth and sixth. Okay. Now, here how many rings are there? 1, 2, 3, 4, 5, 6, 7. So, 7 secondary xylem rings. Okay. The deposited in the TS of the stem, when we see the deposition of the secondary xylem, each occurs in the form of a ring when we see the TS of a stem. So, each ring will indicate the age of the plant. So, that wise we called it as annual ring. In 1 year, total formation of secondary xylem in the stem. When we see the TS of the stem, you will see the rings. Normally, when we cut the wood also, you will see the rings in the center with the pith. Okay. So, each ring will indicate, if there are 7 rings, then you can say the age of the plant is 7 years old. right. So, in this case this is the activity of the cambium, this is formed by the activity of the cambium. The secondary xylem is formed which shows in the form of ring, those are called as annual rings. Now, in case of temperate and tropical plants, the variation in the activity of the cambium varies depending upon the season. Now, in case of tropical and subtropical climate also, the activity of the cambium changes, and here we will talk in terms of in temperate, tropical, subtropical regions, the activity of the cambium varies depending upon the season. Now, here we talk about two terms, those are called as early wood, late wood, or we can also call it as spring wood, autumn wood. These are important. These are the terms we are supposed to study here. Now, first in reference we will talk about the early wood or the spring wood. Okay. Now, what happening here in the spring season, when the activity of the cambium is occurring, the xylem vessels will be thin and longer. Okay. The xylem vessels in the early wood will be thin and longer, and as this wood formation or cambial activity is occurring in the spring season, in favorable season, those are called early formation is occurring here, those are called as early wood or spring wood. And if the activity of the cambium occurs in unfavorable condition, so what is the unfavorable condition here? That is called as winter season or winter. The unfavorable season, that is called as winter. So, the wood that is formed is called as late wood. The wood that is formed is called as late wood, and it is also called as autumn wood. Here because of the activity of the cambium, the xylem vessels which are formed here are narrower and much thicker when comparing to the early wood. So, we are supposed to talk in terms of two definitions here, that is spring wood and the winter wood. So, spring wood is a type of wood which where the activity of the cambium is occurring during the spring season, where the division of the cambium leads to the formation of xylem vessels which are much thinner and longer, whereas division or activity of cambium when it is occurring in unfavorable season, here the formation of wood is in a latter condition. So, we call it as late wood or autumn wood. Here the condition of xylem vessels are thicker and narrower. Okay. Now, ultimately these both will form a continuous ring. Okay. Now, when we are seeing in the TS of in certain plants, the early wood vessels are very large and clearly differentiated from the autumn wood. Okay. Next, we are supposed to study about two more terminologies, those are called as as we said, the xylem vessels which are thin and longer are seen in the early wood. Next, the xylem vessels which are narrower and thicker are seen in the late wood. So, what is the difference between diffuse wood and diffuse porous wood and ring porous wood? Diffuse porous woods are those type of woods which have the appearance of the vessels clearly. So, then that type of wood with a vessels which is thinner and longer, which will show the clear appearance of a vessel, a type of wood which will show the clear appearance of the vessel, then that is called as diffuse porous wood. Example for this will be quercus. Now, ring porous wood, in some plants there is no clear demarcation, or we cannot see the clear occurrence of the xylem vessel or vessel elements, as we have seen in the diffuse porous wood. There will be no clear demarcation between the xylem vessel elements. So, that type of wood is called as ring porous wood. Example for this will be eucalyptus. Okay. Now, here this may again this will lead to the formation of all this circumstance continuity will lead to the formation of annual rings. So every year there is a formation of ring of secondary xylem. So each ring is called as annual ring, and total number of annual rings will count the age of that particular plant. So what are these annual ring? Basically these are nothing but the formation of secondary xylem per year. So now there is a word called as dendrochronology. There is a word called as dendrochronology. What is dendrochronology? Here it is nothing but estimation, or we can say the study of the age of the plant, taking into consideration the number of annual rings present in a plant, is called as dendrochronology. What is dendrochronology? Estimating the age of a particular plant, taking into consideration the n number of annual rings it is having. Okay. If we have 50 annual rings, the age of the plant will be 50. If you have 200 annual rings, the age of the plant is 200 years old. Okay. The study which will dig into detailed study of n number of annual rings in a particular plant and give us the age of a particular plant, then that is called as dendrochronology. Okay. Next, we are supposed to talk here one more two terms. Those are called as sapwood, heartwood. So what is sapwood and what is heartwood? For example, after few growths of regular continuous secondary growth occurring, what will happen? The early form wood, which is the earlier form wood because of the secondary growth, then that will become darker and move towards the center, and the latter form wood will be growing. Because of after few years, because of the continuous growth, the early form wood will come towards the center and the remaining will be the newly formed wood. So this continuity, because of which the early form wood will become darker and will become immature, it will become inactive and it will become dead. So this type of portion of wood is called as, heartwood. Okay. Okay, this heartwood, because it is in the center of it is called as heartwood. Now, in addition to the heartwood, you will see a small protrusions, or those are called as protrusions, or we can small holes or some passages like that. Those are called as tyloses. So, what is the basic function of tyloses here? This tyloses will abstract the entry of solution into the plant body, and it will also not allow the excess, or it will stop the growth of fungal mycelium, where it is a possibility of occurrence of fungal mycelium in the vessels of the secondary xylem. So ultimately this type of tyloses will be present in almost all the angiospermic families, and this type of tyloses are seen in the mature wood. So what is the use of tyloses here? The tyloses will help in increasing the age of the mature wood. The mature wood xylem vessels are filled with gums, resins, pigmentation, coloring compounds, all that. So now the transport of different food materials occur in the secondary xylem tissue. So this transport of the material is occurring in the secondary xylem tissue. So we can say that this secondary xylem tissue will be active in nature, and these are lighter in color. So the color of this area will be not in the center, the area surrounding the center will be lighter and pink in color. So this type of wood, which is active in nature and helps in the transport of food material, is called as sapwood. Okay. Now, as the secondary growth goes on, there will be continuous formation of heartwood, and there is a continuous formation of sapwood going on. So clearly we can demarcate or differentiate between the sapwood and heartwood because of the presence or because of the appearance of their color. As we said, the heartwood is darker in color and it is a dead tissue. It occupies the center of the stem. The heartwood is dark red in color, and the sapwood is the layer surrounding the heartwood, that is light pinkish in color. So the line of demarcation which is present between the sapwood and heartwood can be seen in some plants, and in few plants like PCA you cannot see the demarcation between the light pink and the darker heartwood in color. Okay. So this is between the sapwood and the heartwood. Heartwood is basically dead and inactive, and sapwood is an active which helps in the transportation of the food material. What are tyloses? Tyloses are the protrusions which are developed in the xylem tissue and which are helpful in the stoppage of the transport of solutions, and it is also stopping the fungal mycelial growth in the xylem elements, and it is helpful in the increasing the age of the mature wood. Okay, next we will talk about secondary phloem and secondary xylem. Next thing we are supposed to study under secondary growth is, we have studied about secondary xylem, vascular cambium, cambial rings, annual rings. We have talked about fascicular cambium, intrafascicular cambium, interfascicular cambium. Now we will talk about secondary phloem. What is secondary phloem? It is more or less similar to that of the primary phloem. It has all the elements like sieve elements, companion cells, axial parenchyma or phloem parenchyma, and we have phloem fibers here. Here the sieve tubes are much larger here, and these are staying for the longer time here. The sieve elements which are present in the secondary phloem helps in the translocation of the food material. In the similar way, the companion cells are also helpful in the translocation or movement of organic solutes in the secondary phloem. Next is axial parenchyma. All the inorganic crystals and the reserve starch material, food material which is present, is stored in the parenchyma cells. Next, the fibers which are present in the phloem are called as in the secondary phloem tissue. The fibers are called as the bast fibers, which are commercially important. When we talked about the phloem tissue, we talked in detail about bast fibers. These are the fibers which are commercially used in the making of ropes and mats. Now next we will talk about the secondary xylem. Now in the case of secondary xylem, we will study in this secondary xylem is divided on the basis of the type of division of cells it is occurring. There are two types of systems here. Those are called as axial system and ray system. In axial system, the division of the cells is vertical. It has all the xylem elements which are present. Those are xylem parenchyma, xylem vessels, xylem initials, xylem fibers, all those are present. The division is vertical in section, and here in ray system the division of the cells is occurring radially. Next we will talk about the secondary xylem. When we are talking about secondary xylem, here the secondary xylem is divided into two directions. One is called as axial system type of division, next is ray system. In axial system, the division of the secondary xylem tissue is vertical, and it is almost parallel to the axis of the stem. So here in this axial system, we will see the different elements of xylem present. We will see the presence of xylem tracheids, xylem vessels, xylem fibers, and also the xylem parenchyma. And next, when we come to the ray system of the division of secondary xylem, in ray system you will see the division of secondary xylem in the radial pattern, and this is almost at right angles to the axis of the stem. The cells which are undergoing the radial division are at right angles to the axis of the stem, and here you will see the presence of medullary rays, and because of this existence of medullary rays and this type of radial division, the secondary xylem is connected with the pith, cortex and phloem tissue. Okay. Now next we will talk about we have talked about the cambium, primary phloem, cortex, protoxylem. Now we will talk about the structure called as periderm. Vascular tissue system. Here we will talk about vascular tissues. Okay, now here in vascular tissues, due to the secondary growth, what is happening? The area of vascular tissue system is increasing, and this will lead to the increase of the area of the vascular tissue system, and because of which there are stretching and pulling of all the epidermal cells. So at the initial stage, what will happen? Because of the secondary growth, the epidermis stretches, increases its width area, and slowly after stretching, as the secondary growth goes on continuing, the epidermal cells break. Okay, first they stretch and automatically they break. So what will happen? Because of the breakage of epidermis, the internal cortex cells which are there, which will be exposed to the outer environment, which may not be very good for the growth of the cortex cells. So what will happen? To protect these inner cortex cells from the outer exposure of the atmosphere, a type of epidermal cells tissue develops. Those are called as periderm. Okay. Now what actually I said here is, as the secondary growth occurs, the vascular region or vascular tissue is increasing in size, the vascular tissue system or vascular bundles, that all will increase, because of which there is a tremendous amount of pressure existing, the internal structure will goes on increasing. So, there will be tremendous pressure exhibited on the epidermal cells. So, because of the secondary growth is continuously occurring, what will happen, the slowly the epidermal cells will be normal size. As the secondary growth occurs, they will expand, and because of the excess pressure they will burst or they will break up. And what will happen, if they are breaking, this epidermal tissue system is there, this will burst, and this will burst, and this will lead to the exposure of internal cortex tissue. So, this internal cortex tissue will come directly in the contact with the outer atmosphere. So, to protect that, what will happen, a layer of similar to that of the epidermal cells, a layer is developed, that is called as peridermis. Now, peridermis, when we talk in detail, it is of three types, that is phloem, phloem and phloderm. Phloem, cock, this is developed, phloem is developed from the cock, phloem is developed from the cock cambium, and this is developed from the secondary cortex. Okay. At intermediate level you have this much only, like periderm is a layer which is developed in the replacement of the epidermis when the breakage of epidermis occurs because of the excessive pressure created by the continuous secondary growth on the vascular tissues or internal tissues. Now, it is of three types, phloem, phloem and phloderm, that is phloem is of the cock and phloem is of the cock cambium, and phloderm is originating from the secondary cortex. Okay. This is about periderm. Next we have lenticels. Before lenticels we will talk about the bark. Next topic we have is like bark. What is bark here? During secondary growth, what will happen? Because of the secondary growth, the epidermis is replaced by the peridermis, and the cock cells which are dead are unable to transport the food material to the outer regions of the cortex, which are toward the peripheral regions of the cortex. What will happen? All these cock cells will become accumulated. The cock cells are dead cells which will not allow the transportation of the food material from inner to the outer regions of the tissues are stopped. So, these all cock cells which are dead, and along with those cells which are not able to transport the food material to the outer regions will become dead and accumulate and lead to the formation of a structure called as bark. And this bark is of different types. It may be either in the form of continuous ring, or it may be in the form of strips, or it may be in the form of scales, or it may be in the form of small patches. So, this is about the bark. Okay. Bark which is present is nothing but a collaborative work of dead cells and the cock tissues which are present, which are not helpful in the transportation of material. So, now here this cock cambium, if the thickness of the bark depends upon the origin or the position of the cock cambium which is occurring. If the cock cambium is occurring towards the inner side, the bark will be thinner, and if the origin or the position of the cock cambium is towards the outer side, then the bark will be thicker in position. Now, the cock cambium or bark occurs in different forms, like if it is in the form of a complete ring, that bark is called as ring bark. Example, you can see in Magnifera indica and vitis. And if it is in the form of an overlapping strips, or the strips parallel strips will be present, it will have in the form of strips which are overlapping to each other, then that type of bark is called as scaly bark. Example, eucalyptus. This is about your bark. Next we will talk about lenticels. Now, what are lenticels here? In secondary growth, you will see the presence of cock cambium. So, what is happening here? Cock cells or cock cambium cells or the dead cells which are closely attached to each other, are very closely arranged to each other, and there is no intercellular spaces in between them, because of which what will happen, this the transport or exchange of gaseous material or exchange of liquid or solution in between the two different cells is not possible. So, how the exchange of gases between the internal and external environment takes place? In such condition where the cock cambium is existing, so the epidermis will become periderm, and there is no possibility of exchange of material between internal tissue towards the outer atmosphere. So what will happen there, at this stage you will develop a sandy soil like structure, soil colored, spotted like small patches like cells, which will help in this exchange of material, and this will help in the gaseous exchange, and this will occur below the stomata, or we can say these type of lenticels are present opposite to the stomatal cavity, opposite to the stomatas, and almost work as the stomata and helpful in the exchanges of the gaseous material. So these lenticels are present opposite to the stomata, and these are very near to the substomatal cavity. More or less the exchange of gas material when there is an occurrence of the coccambium or secondary growth or peridome, so which is stopped, that can be continued by the presence of lenticels.